Hello friends, I'm Dr. Bharat Jagyasi and today we are going to talk on understanding resistant patterns of multiple drug resistant organisms, and we are mainly going to talk on the acquired resistant pattern because in the initial session we had already spoken on the intrinsic resistance patterns. And allow me to share my PPT to start the proceedings. So in this whole talk, we are going to have a comprehensive analysis of different resistant mechanisms, identification approaches, and targeted treatment strategies for critical care specialists. So top MDROs in critical care, what we all can think of is carbapenem resistant Enterobacteriaceae, MRSA, MDR, Pseudomonas, and MDR Acinetobacter baumannii. Escape are the pathogens critical 6, and these days it's been critical 7, that is escape E also has been added. So it could be the Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumonia, Enterobacter baumannii, Pseudomonas aeruginosa, Enterobacter species, and the extra E which has been added is covers E. coli. In the initial talk, we had spoken about the lactose fermenters versus non-lactose fermenters. So lactose fermenters, we all know, appears as pink red colonies on Mackenzie agar due to acid production, and these consist of E. coli, Klebsiella pneumonia, Enterobacter species. Non-lactose fermenters appears colorless or pale colonies on Mackenzie agar, and these consist of the Pseudomonas aeruginosa, Acinetobacter, Stenotrophomonas multifida. These are more malignant organisms with more intrinsic resistant mechanisms available, and also as they evolve, they acquire many different resistant mechanisms. Maybe they come up with the multiple resistant mechanisms at one go. So when he spoke about the resistant mechanisms, we already spoken about this. There's something called as intrinsic resistance, and also we have the acquired resistance. So in this talk, we're going to mainly talk on the acquired resistance, which goes from mutations or gene transfer and driven by the antibiotic pressure. That the more the exposure of antibiotics, more is the evolution of the organism. So understanding the evolution of antimicrobial resistance, so we have genetic basis, that is foundational mechanism of resistance development in bacterial genomes. Transfer mechanisms, as we all know, different methods available: transformation, transduction, conjugation, that is horizontal genes transfer, and the resistance strategies. Those are specific to the cellular mechanisms employed to neutralize antimicrobials, and this is what is important as we spoke further. So when we talk about the mutational resistance, we see one in approximately 107 to 109 bacterial cells create point mutations in drug target genes and alteration in the related regulatory sequences. When we talk of the horizontal gene transfer, that is nature's resistance sharing network. So we have the transformation where the direct uptake and incorporation of naked DNA from the environment happens. Transduction, which is bacteriophage mediated transfer of DNA between the bacteria. And conjugation, that is direct cell to cell transfer via specialized pili, which is also called as the bacterial sex. But what is important is mechanism of antibiotic resistance in terms of the drug. So if we have to classify that, it would be the drug uptake limitation, drug efflux, where the drug is thrown out of the cell, drug inactivation, where they render or produce many enzymes which render our neutralizes our antimicrobials, and also the drug target modifications. By that way, drug are made useful and inactive as far as the killing of the organism is concerned. So limiting drug uptake is a permeability barrier. There's an alteration in the porine proteins that reduce antibiotic entry and changes in lipopolysaccharide composition and formation of protective biofilms, creating physical barriers. When you talk of the drug efflux systems, there's a specialized protein complex pen the cell membrane, actually pump antibiotics out of the cell before they can reach the targets. And this range from narrow spectrum pumps specific to one antibiotic class to multiple drug resistant pumps that export diverse compounds. And overexpression of efflux systems in organisms like Pseudomonas and E. coli confers resistance to multiple antibiotic classes simultaneously. Drug inactivation, this is what we are going to understand and read in detail, and it has quite a bit of the clinical implications as well. We'll discuss as we go further. So these can produce beta-lactamases, which hydrolyze the beta-lactam ring in penicillin, cephalosporin, carbapenem, aminoglycoside modifying agents, chloramphenicol, acetyl transferases, carbapenemases, and cephalosporinases. The last mechanism is D, that is about the drug target modification. It could be the ribosomal alteration as seen in aminoglycosides and macrolides. PBP modifications uh, could be seen by different organisms, maybe the E. coli, as we discussed further. Enzymatic bypasses, they come up with the alternative metabolic pathways, and the target protection that is a protein that specifically shield the target from antibiotic binding. So what we really interested are in the drug inactivation enzymes, and we talk of it. We have the Ambler classification of beta-lactamases, where the beta-lactamases are classified as class A, B, C, and D. So class A mainly consists of ESBL and KBCs, and class B is metallobeta-lactamases. Those are the dangerous classes where even the carbapenems are not effective, and there's a rationale of new antibiotics coming in. Amber class C covers the MPCs, and class D we have the OXA 48 and 23. So class A, we see more of the ESBL and KPCs and inhibited by the clavulanic acid and tazobactam and avibactam. Class B, what we look into the NDM, not inhibited by the standard inhibitors. Class C mostly covers Enterobacter, Serratia, Citrobacter, and Cefepime carbapenems are the preferred choices. Class D, we have the OXA 48, mostly seen in Acinetobacter and Klebsiella, and they have the variable carbapenem resistant patterns. So if you look at this, when it comes to the ESBL and OXA, they are sensitive to the cefamycin, that is cefoxitim, and when it comes to the MC, they are not really sensitive to, or maybe the partially sensitive to 4th generation cephalosporin, and this is what the differentiation between the ESBL and MC is. So beta-lactamases can also be classified as serine beta-lactamases and metallobeta-lactamases. So serine beta-lactamases are Ambler class A, C, and D, and the metallobeta-lactamases are the Ambler class B. So it could be again class A could be classified as narrow spectrum ESBL carbapenemases with varied resistance to the beta-lactam inhibitors. And class C cephalosporinases MC, again it has the varied sensitivity to the beta-lactam inhibitors. Class D, that is OXA 48, that is where only the avibactam is effective. When it comes to the metallobeta-lactamases, Amber class B, these are carbapenemases and no beta-lactam inhibitor acts over here. Here we have to use different combinations and we'll come through this. So this is overall resistant pattern is stairway to the doom, that is towards death. So we have the beta-lactamases followed by the cephalosporinases, ESBLs, MC, KPCs, OXA-As, and metallobeta-lactamases, which has very high mortality of 50 to 70% as is being reported in different countries. So when it comes to the comparison of ESBL versus MC, it has very critical difference. ESBL is mostly seen in E. coli and Klebsiella, and MC produces Enterobacter, Citrobacter, and Serratia. BL-BLI may work in ESBL in low inoculum in the immunocompetent patient, but they do not work very well in the MC. When it's MC, we have to consider more of the carbapenem. So BL-BLI, where it works in ESBL, so when we have the mild to moderate infection, when the patient is immunocompetent, and especially when it's a urinary tract infection, SSTIs, or intra-abdominal infection where the source control is very good, and organism is E. coli. Of course, when the MIC to the piperacillin tazobactam breakpoint is less than 8, considering UCAS breakpoint, we can consider using BL-BLI. But when it's a severe case, immunocompromised, bloodstream infection, it's Klebsiella enterobacter, and when the MIC is more than 60, considering UCAS breakpoint, we have to consider carbapenem in such cases. When we look into the various antibiotics, so when you talk of the aztreonam avibactam, so aztreonam avibactam is a combination which has been used. The rationally I will discuss in the next few minutes. So it can be used in KPCs, NDM, and OXA 48, and also in Stenotrophomonas maltophilia. Ceftazidime avibactam could be used in KPCs, OXA 48. Fosfomycin may or may not act against KPCs, NDM, OXA 48, and carbapenem resistant Pseudomonas. Polymyxins are partially or may not act against a different group of organisms. Tigecycline, we all know, we learned in the previous talk, those are intrinsically resistant to the Pseudomonas aeruginosa and can be tried in KPC, NDM, OXA 48, and Stenotrophomonas maltophilia. So why this rationale of aztreonam and NDM complex relationship? So aztreonam are resistant to hydrolysis by NDM enzyme. So they can be very well used when the resistant mechanism is NDM, but they should remain active against the NDM producing organism. The problem here is MDM bacteria also carry other beta-lactamases, that is ESBLs and MC, and these enzymes can destroy aztreonam and making it ineffective. This is why the solution is we use combination of aztreonam and avibactam. So what avibactam does is it blocks ESBL and MC, protecting aztreonam from destruction, and which is very much resistant to hydrolysis by NDM enzymes, and it works very well. So how do we do it? Treatment options in carbapenemase. So if it's a metallo-beta-lactamases, maybe the resistant mechanism is NDM, WIMP or IMP. We use prolonged infusion of ceftazidime, avibactam, and astronome. Both infuse at the same time. And when it's KPCs, we use prolonged infusion only of ceftazidime. So when you talk of the real life scenario, so enzyme impact, so when it's class A and we have ESBL, Klebsiella, maybe patient who has a VAP, so those are resistant to the third generation cephalosporin. Piperacillin tazobactam, that is BL-BLA, may fail for the severe infection as standard dose, as we understood. So consider carbapenem. When it's NDM that, and the organism is Klebsiella, patient is in septic shock, those are resistant to all beta-lactams, including carbapenem. So what we can do is we can use a combination of astronome plus avibactam. If those are not available, non-effective, and then maybe we can go and try other methods using tricycline fosfomycin combinations. AmpC producing enterobacter, straight away consider carbapenem. Class D, OXA48, they have variable carbapenem resistant mechanisms, more of meropenem than imipenem, and consider using ceftazidime, avibactam, or fosfomycin in such cases. So when it comes to NDM, are E. coli and Klebsiella same? No, there are different challenges. So as we look into this, so there's something called as PBP, that is penicillin binding proteins, and PBP3 mutations are seen more often in the E. coli. So they reduce the beta-lactam binding affinity, and which is associated with reduced susceptibility. So what it does is reduce astronome binding ability, which is seen in E. coli, leading to higher MIC despite we use a combination therapy, and results in unpredictable clinical outcomes, which is not true when it comes to the Klebsiella because it has less reliance on the PBP3 modifications. And there we see more consistent effectiveness of the combination therapy. So how do we identify it? Either we do the molecular sequencing, that is whole genome or targeted PCR, or also we can do it from the MIC interpretation. So if the astronome MIC is more than 4 nanogram per ml, despite protection, it tells us there's a reduced efficacy of the combination therapy, and this could be maybe the PBP3 mechanism playing over here. So how do we detect different resistant mechanisms? Either we use the phenotypic standard methods by culturing it, looking into the MIC's value sensitivity, or we can also do the molecular methods, PCR for the specific resistant genes, multiplex PCR panels, whole genome sequences, or the multi-TAF TAF for the carbapenemase detection. So this is the NDM infection algorithm. So decision algorithm, identify the organism whether it's E. coli or Klebsiella with carbapenem resistant. And when the moment you see there's a carbapenem resistant, of course it's an NDM suspect NDM production. Check resistance marker, maybe NDM gene detection or coexisting ESBL and MC. Interpret astronome MIC. If MIC is less than 4 microgram per ml, astronome is likely to be effective. If MIC is more than 4 microgram per ml, suspect PBP3 changes, especially in case of E. coli. And adjust based on organisms. If E. coli, then consider PBP3 issues. If it's Klebsiella, then you can very safely use astronome and avibactam combination standard. So how do we decode our culture report? Identify organisms, check resistant patterns, analyze MIC, and also look for resistant markers to guide your therapy. If we look at this small cheat chart or cheat slide, right bug drug combination. So Pseudomonas aeruginosa are inherently resistant to tetracyclines and cotrimoxazole. Ceftazidime avibactam, poor activity against anaerobes, MSSA, MRSA, and Acinetobacter. Quinolones, levofloxacin, ciprofloxacin have limited anaerobic coverage. Moxifloxacin, excellent anaerobic coverage. Usually resistant to colistin is seen in case of Legionella, Brucella, Burkholderia, Proteus, Serratia, and Bacteria fragilis. Novel antibiotics. So when you talk of the ceftazidime avibactam, it covers a very good spectrum of KPCs, OXA48, but the resistance might emerge during the therapy, and we have to monitor very closely. Meropenem, Weberbactam are very good against the KPCs, and but limited real-world data, especially in the Indian population. Imipenem relibactam has very good spectrum against KPCs, maybe against some Pseudomonas aeruginosa as well, and has lower nephrotoxicity as compared to the colistin, but it can be very well considered for the MDR Pseudomonas aeruginosa. Irvacycline is useful in CRAs and Acinetobacter, but not for the Pseudomonas aeruginosa. Plasmomycin most aminoglycoside resistant GNB, it works against the most aminoglycoside resistant GNB, and the dosing is once a day. So thank you very much. Grateful for your attention and time. And as we go ahead, we'll speak further about the different topics, including this antibiotic resistant patterns. Thanks for listening. Please follow the YouTube channel. Subscribe. Thank you.